Hi students, now in this video I am going to explain what is the C4 cycle, what is its importance and how it will be. The detail I am going to explain because people are getting doubts in this one only and up to now three questions given from this one. This is the C4 plant. Now we will see why the C4 plants are adopted. In this one neat question is there and what is its importance. And the next it is what is it is adapted for what purpose, and for this purpose anatomy also it is to show some peculiar anatomy which is called fringe anatomy, that also I will discuss. And the next, whenever we are seeing that is crange of the H-slack cycle, the who are the discovered this one, that is the cyclic one. And in this one, after fixing of the carbon dioxide in the mesophyll cells, in the form of it will fix in the bicarbonate. That is also important, need to remember it. And next, after fixing, it will become oxaloacetic acid 4 carbon compound, and then it will go to the malic acid or aspartic textbook has given. Why? What is this one? What is that one? Where it will be? What is the why it is coming like that? That also you will get the doubt. I will clarify that thing also. And this oxaloacetic acid, whatever the malic acid that it forms, malic or aspartic acid will be sent into the bundle sheath cells where it will be leaving the carbon dioxide. That also important. And where the primary carbon dioxide, primary acceptor, where it is working in the cytoplasm or the stroma, or where it is. All these things detailedly I will discuss. And here and there I will add all the which type of the question can be asked in the NEET, that also I will ask. I will detailedly discuss in this one. So please watch this video properly. And now the thing is, C4 plants already in the skeleton cycle. What are the C4 plants he has given? That you know. The C4 plants are that they show the C4 pathway. Means primary carbon acceptor 3 carbon compound, and it stable product is oxaloacetic acid which is 4 carbon, so we are calling it a C4. Now the C4 plant adapted to dry tropical region. What is this? Dry tropical region means here the temperature will be high because of the high sunlight, and by the adaptation they are tolerating that temperature and high sunlight and showing more productivity. How it is that we will see when the primary carbon dioxide fixation, there I will discuss that one. So but the productivity is high compared to C3 plants at the high intensity of the light and temperature. This is the neat bit. Why it is, that we will discuss in the cycle. Now in this one, for this, what is the purpose of this one? This way they are adopted to avoid the photorespiration. Photorespiration is a waste. What is its importance? Up to now we don't know much, but photorespiration I will discuss in the next video detailedly with factors affecting the photosynthesis. That also important one. In the next video I will show. Now for this purpose, when they are adapting, what they will do, they will show the internal, means where the photosynthesis will be taken in the leaf mesophyll, and in the mesophyll when they are taking the photosynthesis, they will show some type of the arrangement in the mesophyll also. Means whatever the mesophyll means, from the upper epidermis and lower epidermis, whatever is present in between them, that is called as the mesophyll. And in the C4 plants, the particular anatomy can be shown, that is called as crange anatomy. What is that one? Means crange anatomy means, here you see, this is the vascular bundle. Around the vascular bundle, there are the big cells large cells present in the multi-layer. It may be. And it appears like a garland width. That is we can see width. Garland means big size cells. Remember the point. Compared to mesophyll cells, they are the big size cells which are present around the vascular bundle of the leaf inside the leaf. If you find that one in the transfer vertical section of the leaf, that plant belongs to the C4. This is the need bit. And next, how the cells are? The cells are large number of the chloroplasts, and that chloroplasts also they are the having the only whatever we are stroma lamellae. They won't have that. I will discuss in the cycle. And next is the cells used to around the cell thick wall is to present cell wall, and it is impermeable, means it is not allowing gases exchange cannot be taken place through the cells walls of, he will tell, that is the mesophyll, the bundle sheath cells. That is the one more important point. Now the cells, why the and they are not having the intracellular spaces, means here why, what is the reason? Here the thing is oxygen should not enter into the bundle sheath cells. For that one, without intracellular spaces, they used to present at the around the this vascular bundle. And here, so there is no chance of the entry of the any gas into the bundle sheath cells. This is important point. And not only oxygen, but also carbon dioxide also, because he will give the bits in the multi statements also, this one. Now, how it appears like a garland around the vascular bundle. Now, whenever we are seeing the examples, whatever the maize, sorghum, sugarcane, such type of the plants, and in this one, only they means majority or the 80% of the C4 plants belongs to the monocots. Overall, 3 lakhs of the plants belongs to the flowering plants. Out of that one, only 3% only will show. This is out of, but the you remember it, because very less plants which are growing in the tropical region. What is the tropical region? What is the this thing is, when the sunlight is intensity is high, temperature also will increase. So here the chloroplast behavior will be different. Already alignment we had seen in the chloroplast. That not now the thing is to get the when they are getting high intensity of the light. All of you know that the dark reaction and the light reaction. Light reaction will be taken place in the stroma lamellae and the grana. In the stroma lamellae and grana, whatever the enzymes are very less enzymes used to participate, they are the having the tolerance to the high intensity of the light and heat. When the dark reaction, whenever we are seeing, they are the sensitive to the heat, the temperature. So their working efficiency will be decreased in the dark reaction, efficiency will be increased in the light reaction. So what is the problem? Because of that one, the oxygen production will be more, because where the oxygen will be produced in the light reaction, that also in the thylakoid, that already you know. So the production is more, and that must be used in the dark reaction. But dark reaction becomes slow. Naturally, the oxygen concentration will be increased in the stroma, and not only in the stroma, but also in the cytoplasm. So in the stroma, when the oxygen concentration is increased, at that time photorespiration will be taken. That photorespiration we will discuss later in the next video. So to avoid that one, here the carbon fixation will be 2 places, it will be taken place in this one. And this entire thing is explained by the H and Slack scientists as the C4 pathway, that is cyclic. So in this cyclic, whenever we are seeing, you see here carbon dioxide, one more thing, important thing is the primary acceptor of the carbon dioxide in the mesophyll cells of the mesophyll cells in the C4 plants is that is the PEP, phosphoenol pyruvate. Where it came? This came in the glycolysis also. There I will tell again. So, but the thing is, there is a lot of difference. Mesophyll cells of the C3 plants will fix the carbon dioxide in the gas form. Remember, this is important. But in the C4 plants, it will fix the carbon dioxide in the form of the bicarbonate. There a lot of reason is there, because when the light intensity and temperature is high, and you already in the plant growth plant growth and development and plant development growth and development, there you might have you studied already, or you will study. There he has given abscisic acid, which is the negative regulation, the stress hormone also. It will, what in the when the temperature is high, water loss will be more through the stomata. There he has given the textbook clearly, that is, it will close the stomata. It will, abscisic acid will close the stomata. So there is no question of carbon dioxide coming inside. So what? How the C4 plants are getting? You will get the doubt. The thing is, here remember and listen properly. The carbon dioxide, even though stomata are closed in the high temperatures and high intensity of the light, the carbon dioxide will be permeable through the cuticle layer, which is to present on the upper surface of the leaf, but it is less. Understanding, no? So when it is less, slowly it is coming through the cuticle layer into the mesophyll cells. But here the plant will play one important role, that is, whenever it is entering into the cytoplasm is having, this is not out of the, but the thing is, it is important. Why it is the reason I am telling. It will have the that is a carboxylic anhydrase, carboxylic anhydrase enzyme used to present in the C4 plants in the cytoplasm. What is carboxylic anhydrase? Go and see the whatever the biomolecules in the enzymes he has given. It will increase the absorption of the carbon dioxide millions of the times. That he has given clearly, you see. So that he has not given here, but in the references you can find out. So that will speedily it will absorb, even though it is coming slow, but absorption will be very fast, so it will come very fast. So carbon dioxide will be taken in the cytoplasm through the carboxylic anhydrase. That is out of, but for reference I am telling. Now the thing is, when it is absorbed, PEP used to present in the cytoplasm. Remember it. PEP, phosphoenol pyruvate, which is the primary carbon dioxide acceptor in the C4 plants. It is to present in the mesophyll cytoplasm. And one more enzyme, PEP carboxylase, which is to fix the here. It entered, carbon dioxide entered into the mesophyll cell, and it will be dissolved in the dissolved condition. It will formed because of the, already I told, that the carboxylic anhydrase. It will be dissolved and forms the HCO3. But there he has given, that is the carboxylic acid. But one hydrogen it will lose and forms HCO3, which is the bicarbonate alkali. Because acid is not good for the cytoplasm. So this bicarbonate, and next is the bicarbonate and phosphoenol PEP carboxylase and carbon dioxide water will form the HCO bicarbonate, and both they will fuse they will be unite by because of the PEP carboxylase. It will take the bicarbonate and it forms the oxaloacetic acid, which is the 4 carbon, and formation oxaloacetic acid will be in the cytoplasm, and formation and observe the primary acceptor are used to present in the cytoplasm. Remember all the points. These are the important things, but indirectly it told there in our textbook. Now, so oxaloacetic acid 4 carbon compound, it will be converted into malic acid or aspartic acid as given in the textbook. Why? What is the reason? Malic acid, now he is discussing about the how the carbon dioxide is taken in the C4 plants in the mesophyll cells. Here, one important point you should remember, that is malic acid, whatever that thing is, here the malic acid formation will be done by taking the NADPH. NADPH is forming, but primary acceptor of the carbon dioxide in the stroma is differing. So that light reaction, that a non-cyclic is going on very fast. So it will take that one and it will be converted into malic acid. If you want any information, go and see in that Krebs cycle. Malic acid is converting to oxaloacetic acid by giving out one molecule of NADH. NADP and NADH both are the almost same, that I will discuss there. So here that NADPH will be taken by the formation of the malic acid in the oxaloacetic. Oxaloacetic acid will be converted into the malic acid by taking of the NADPH, but not textbook, he has not given. So okay, you read it like this one. And aspartic acid. Why aspartic acid? When the NADPH is not available, means much it is going very fast because it is absorbing because of the carbonic anhydrase. It is absorbing very fast. So NADP, whatever the NADPH formation in the non-cyclic in the Ghana, if it become late, it will be converted into the aspartic acid. Aspartic acid not required for the oxaloacetic acid, aspartic acid, it is not necessary to take the whatever the NADPH. So when the NADPH is not available, aspartic acid will form. When NADPH is present, it is available, it will form the malic acid. Remember the point. For your information, this is extra information I am giving, but you will get the doubt. For that one I am telling all the things. Now the thing is here, when the malic acid or aspartic acid, they will travel into the mesophyll cells. Mesophyll cells, where the big cells which are present around the vascular bundle, already I discussed this one. Now the thing is here, when the mesophyll cells, when they entered through the plasmodesmata, means the cytoplasm connections used to present in the cell to cell through the plasmodesmata. So through that one, it will come into the mesophyll cells, bundle sheath cells. So and one more thing, whenever he is drawn in the textbook, that is wrongly he has given, because whatever the bundle sheath cell size is big, he is telling, but he has drawn both are equal size. Such type of mistakes is common in the NCERT book. So here oxaloacetic acid will become malic acid or aspartic acid and transported through plasmodesmata to bundle sheath cells. And next, after reaching into the bundle sheath cells, it will be decarboxylated. Then carbon dioxide will be released. Carbon dioxide will be released by decarboxylation. Remember, it has taken in the liquid form but released in the gas form. That also he may twisted, he may ask. and this carbon dioxide will enter into the C3 cycle in the chloroplast. Remember, the releasing decarboxylation also will be taken in the cytoplasm, not in the chloroplast. But that is also important. So fixing will be in the cytoplasm and releasing also in cytoplasm of the bundle sheath cell. These are the important points. And one more, this is there that I will explain lastly. So carbon dioxide will be relieved and it will go to the stroma lamellae, because that is also different. That I will tell later on. But the entire thing, unnecessary things, it will become. For that one, I am not discussing. So carbon dioxide in the gas form, it will enter into the C3 cycle, which is present in the bundle sheath cell. So remember, by the C4 cycle, by C4 will be taken, means fixation of the primarily carbon dioxide will be fixed in the 4 carbon compound in the mesophyll cell, and released carbon dioxide in the bundle sheath cell, and here the C3 cycle, Kelvin cycle will be taken. So Kelvin cycle will be in the bundle sheath cell, and C4 cycle absorption, C4 we are calling, it will be in the mesophyll cell of the C4 branch. Where it is, how it is, that he has given in the last 16 points, 17 points he has given. That also I will give the information. Now, after decarboxylation, 4 carbon compound, it is either aspartic acid or the malic acid, both are the 4 carbon compounds. They lost 1 carbon in the form of a carbon dioxide. So it will become 3 carbon compound. So 3 carbon compound acid will be formed. What is that one? Pyruvic acid. It will form the pyruvic acid, which is the end product of the glycolysis. Here the pyruvic acid will be formed. This pyruvic acid will be transported into the mesophyll cells through the plasmodesmatagon. So what is the important here? In the glycolysis, you go and see. Phosphoenol pyruvate will become pyruvic acid. Means phosphoenol pyruvic acid, that is pyruvate or pyruvic acid, that I will do. PEP will become pyruvic acid by giving one ATP, by taking the by giving out the phosphate. But here pyruvic acid, 3 carbon compound, will become the phosphoenol pyruvate, means it required phosphorus. So it, by taking the phosphate, it will become for the phosphoenol pyruvate, and it will in the mesophyll cell. So in this way cyclic it is going. So here one more thing I want to tell you. If it is C4 acid is a malic acid, malic acid will enter into the chloroplast of the mesophyll cell, and it will be taken NADPH, which is produced by the non-cyclic, and it will become oxaloacetic acid will go to the chloroplast, and it will take the NADPH, which is produced in the stroma, and it will become malic acid, and it will come out and it will be transported. Aspartic acid won't do like that. Directly it will go. And the releasing of the carbon dioxide also, aspartic acid directly will release carbon dioxide in the cytoplasm. Malic acid will release carbon dioxide in the chloroplast. So for that only some people are confusing and telling they will take either malic acid cycle or aspartic acid cycle, but it is a difference. It is not necessary to know detailedly, but the concept, if you want to know, this is important. Next, here the thing is, so the cells, these things, bundle sheath cells. Now you see here, carbon dioxide and water, the water will become the bicarbonate, and it will be mixed with the phosphoenol pyruvate and forms the oxaloacetic acid, 4 carbon compound. And where it is in the mesophyll cell, it is forming That also important, where it is forming. And next, oxaloacetic acid will be converted into the malic acid or aspartic acid. Why? The reason I explained. And next, they will be transported through the mesophyll bundle sheath cells through plasmodesmata. This is also important bit, because, and next is plasmodesmata, it will enter, and there the C4 acid means either maybe it may be the malic acid or the aspartic acid in the textbook according to. It will release, it will be decarboxylation will be taken place, and carbon dioxide will be released in the form, what the form that also I discussed already. And next, the 4 carbon compound, by sending out 1 carbon dioxide, it will become 3 carbon compound, that is the 3 carbon acid, that is the pyruvic acid. It is not given in the textbook, only C3 acid it will, okay, leave it. And this one will become PEP, phosphoenol pyruvate, and it will travel, C3 acid will travel into the mesophyll cell through the plasmodesmata. Then it will become PEP, where it will be PEP will be regenerated. He will ask in the mesophyll cell. Remember it. All these are important questions. But any cycle, whenever it is taking, it requires some ATP. Nowhere ATP it has taken, but it will take ATP. That is out of, but old cell, but he is asked sometimes. 12 ATP extra it will take. C4 plants for fixation, for doing for formation of the one glucose molecule, it will take 12 ATP more. Why? Because already you know Kelvin cycle, how many ATP it is taking? 18 ATP only. Because the ATP will be 18 and NADPH will be 12. That already you know. Here extra it will take 12, then it will become 30 ATP will be taken. Now I want to tell why the product will be high. Because there the carbon dioxide directly in the C3 plants, in the mesophyll cell, directly it will enter into the chloroplast and it will enter into the stroma and it will do the dark reaction. But here carbon dioxide is less because of the stomata are closed, why I discussed. And even though they are getting more carbon dioxide by the having carbonic anhydrase enzyme which is present in the cytoplasm, that is out of, but I discussed this one. And because of that one, it is extracting very fast. That how much fast it is, go and see in the biomolecules, in the enzymes. Now it will become bicarbonate. So carbon dioxide availability is less for the C3 plants, but C4 plants are having the adaptation by having that enzyme, and it will take the more carbon dioxide and it will form. And for that only, when the carbon dioxide is high, then naturally it will be more. Next, one more thing. In the bundle sheath cells, whenever you are seeing, here the chloroplast used to have only stromal lamellae. It is having the only stromal lamellae. Means the grana is absent. Why? Because if the grana and everything is there, non-cyclic will be taken. And non-cyclic, whenever it is taken, oxygen will be evolved. So to avoid that one, it is not having the grana. Only stroma lamellae will be there. Stroma lamellae already you know, that is the ATP will be cyclic. ATP, only ATP will be produced. So it is agranal. We used to call it as a agranal chloroplast are present in the bundle sheath cells, and granule and other granule and with the stroma, it is to present in the mesophyll cells. Now one more thing is, what it is separating, you will ask. In the C4 plants, it is separating. One thing remember it. Light reaction and dark reaction. Only dark reaction is separated. Remember it. Light reaction will be taken place here also. Because that is the NADPH is producing. Here also light reaction is taking place. Only cyclic. Remember it. All these are the neat points. And so here NADPH is not produced in the bundle sheath cells, but NADPH will be produced in this one, in the mesophyll cells. One more point is, in the mesophyll cells, why they are not they separated? Because if it is in the C3 cycle, if it is like that, the photo respiration will be taken place because of oxygen, because it is the Robesco enzyme. That what is the Robesco enzyme and its activity we will see in the next video, in the photo respiration. So Robesco used to present in this in the mesophyll bundle sheath cells, and Robesco is absent. Robesco is the main carbon dioxide fixation, primary carbon dioxide fixation enzyme. That in Kelvin cycle, you know. So Robesco is not present in the mesophyll cell. Robesco used to present in the bundle sheath cells. Now the thing is, so here mesophyll cells no Robesco, and in the bundle sheath cells no PEP, because whenever it comes as the malic acid or the aspartic acid. When it is going out as a pyruvic acid. So there is no question of PEP present in the bundle sheath cell. PEP will be regenerated in mesophyll cell. All are bits only. If you listen properly and follows any type of the question, you can answer it. Now, next thing is, he has given 17 points in the ending. What is that one? Kelvin cycle. Kelvin cycle will be taken place in the C3 plants in the mesophyll cells, because there is no bundle sheath cells participation in the C3. But Kelvin cycle will be taken place in the bundle sheath cells of the C4 plants. And Kelvin cycle, primary acceptor what it is, and like that he has asked. Primary acceptor of the C3, it is the whatever we are RUBP, and here it is the PEP. Like that, all the things you can. And first acceptor, first carbon acceptor in the mesophyll means that is the RUBP, that is the RUBP in the C3. In this one PEP, and CO2 fixation as the primary acceptor. Primary acceptor there it is the RUBP. Primary acceptor is the RUBP, and here is the PEP. And cell types fixed carbon dioxide. Only one type will be there in the C3, only mesophyll cell. But carbon dioxide fixation twice in the C4 plants. First it will be fixed in the mesophyll cell, and again it will be released in this cytoplasm of the bundle sheath cells, and again it will be fixed. So 2 times it will be fixed. And number of the primary acceptor carbons, that is the 5 carbon compound RUBP in the C3. In the C3 plants, in the mesophyll cells, RUBP will be present, and it is the 5 carbon compound. And primary acceptor in the C4 plants in the mesophyll cells, that is the PEP phosphoenol pyruvate 3 carbon. And next will be the number of primary acceptor carbons what has formed. 5 carbon compound taken the 1 carbon dioxide, and it become the 2 molecules of phosphoglyceric acid in the C3 plants. But in the C4 plants, it will take the PEP will take, and it will become 4 carbon compound. The splitting is there, here the splitting is not there. 5 carbon compound RUBP will take 1 carbon dioxide and 6 carbon compound, which is the very temporary. It will be splitted into 2-3 carbon compounds, that is the phosphoglyceric acid. This is also need bit. But whereas in the C4 plant, it will take carbon dioxide in the PEP, and immediately it will form only 1 molecule of the oxaloacetic acid. Oxaloacetic acid, again what happened I discussed. And the next will be the number of product number, that is the 4 carbon compound in the C4 primary acceptor after taking, and 3 carbon compound there. and fixation rate at high temperature. At high temperature, fixation rate will be low in the C3 because that is a photorespiration will be taken place. But it is separated in the C4 plant. So it is no effect high temperature, and the other and high temperature is not affected, high intensity of the light is not affected. Next will be the now temperature when dying. One more thing, in the bundle sheet cells are having the thick cell wall, we told they are impervious. Not only that one, it will be useful to protect the heat also, temperature also it will control because of thick wall. And the next is it is not having the intercellular spaces. So and one more thing is where the vascular bundle is to present just in the inside of the mesophyll cell. So the high light intensity will be reduced because here somewhere epidermis, lower epidermis is to present, but it is somehow some cooling. That also important, but it is indirectly he came and asked, but the thing is you have to imagine it. And next is that is the importance of the cranch anatomy. Now I am telling, because if you know this one, then only you will come, because it is not allowing the, even though oxygen concentration is more in the mesophyll cell, it is impervious because of the thick cell wall of the bundle sheath cells. That is the reason this can be asked in the NEET. And next will be the high temperature, and next will be the carbon dioxide. Carbon dioxide concentration increases, there is no effect, but it is decreases, then oxygen will be taken, photorespiration will be taken. So high and low light, high light photorespiration will be taken, low light for no photorespiration. So both will be no photorespiration either in the C3 or C4. C3 and C4 are differing, remember it, in the high light and high temperature only. At that time only the C4 plants are having the advantage. But it will be at only midday, that I will detail discuss in the photorespiration. And next will be the temperature he has asked, given. Means temperature 20 to 25 optimum for the C3. But the plants of the C4 plants, they will be optimum is 30 to 40. So highly tolerated for the light and temperature. Now I am expecting, last year he has not given from this one. So definitely I am expecting this year one question will come from this one in the 26. So this is the about C4 cycle pathway. And one more thing here, you listen it properly, read it properly in the textbook, and textbook only you have to follow. But additional information I given, because you will get the doubt why it is, how it is. For that only I clarified. And then only you will get the concept. Then only it is possible. Because even teaching people also, they are following that one, this one, and they are different ways are there. But I hope it will be useful for you. And if you like this video, please subscribe my channel. Next tomorrow only I am going to release one more thing, that is a photorespiration with the factors affecting the photosynthesis. That also important points are there. That also I will discuss. And after that one, I will enter into the respiration. The respiration also will be, teaching will be like this. I hope you will follow. And if you want to contact me, my number is in the thumbnail, that you can contact me and you can ask your doubts also, no problem. Okay. And next is, if you like the video, please subscribe my channel and like it.